Hello, do you hear me? I'm audible?
Now, yes, Ali. Hi, how are you?
Yes, I am fine. How about you?
I'm fine, Alhamdulillah.
MashaAllah. Okay, let's start today's session. Okay, warm welcome to everyone joining us today. I am Arid Raz and I will be guiding you through this exciting journey into bioinformatics. This is the first session of our bioinformatics workshop series, where we will start from the basics and progressively move toward the advanced topics. Today's session is all about understanding what bioinformatics is, its scope, goals, limitations, emerging trends, and a generic overview of databases. Before we into it, let's take a moment to appreciate how biology and data science have become together to revolutionize fields like genomics, drug discovery, and personalized medicines. Feel free to ask questions, share your thoughts, and actively participate. This workshop is designed to be interactive, so let's make the most of it. Let's begin. Okay, now I'm going to share the screen to all of you. Okay, now please let me know. Do you see the screen? Decoding
Yes,
live.
Ali. Yes.
Okay,
Yes,
okay. Thank you. Okay. Uh, Now we're going to start. Okay, someone want to join, I think so. First of all, admit it. Okay. So actual the whole series is all about the decoding life bioinformatics from beginner to pro. Okay. And the today's session is all about the from DNA to data, introduction to bioinformatics and its applications. Okay, now let's start. Okay. If you can't explain it simply, you don't understand it well enough. Albert Einstein. Okay. Uh, it's an introduction about me. My name is Dalid Ras, and I am a passionate BS genetics and molecular biology scholar at University of Health Sciences, Lahore. Brings a wealth of knowledge and hands-on experience in bioinformatics, stem cell therapy, genomics, genetics, and evolution and molecular diagnostics. And today's is all about the bioinformatics. With a keen interest in advancing the scientific discovery, Ali is dedicated to inspiring others through engaging learning experiences and fostering a deep understanding of cutting-edge molecular biology techniques. Okay, workshop one. From DNA to data. Introduce the fundamentals of bioinformatics and its real-world applications in this session. Familiarize the participants with the key biological databases and tools. Bridge the gap between biology and computational analysis. Equip the learners with the essential skills for data-driven biological research. Lay the foundation for advanced topics in sequence analysis, phylogenetics, and structural biology, empowering the next generation of bioinformaticians. Okay, I am introverted but willing to discuss the bioinformatics is just a quote. for bioinformatics. Okay, the introduction to bioinformatics. Bioinformatics is rapidly growing field that merges the biology, computer science, and mathematics to analyze and interpret the biological data. Okay, the word is biological data. It may be a sequence data. It may be about a DNA. It may be about the protein. It may be about the RNA. It may be about the it can be about the protein structures. Okay, this is actually a biological data that we get by the different techniques. Okay, now at the end we have to analyze and interpret that data. Okay, so in order to analyze and interpret that data, we use bioinformatics. Okay, it plays a crucial role in modern research, helping the scientists understand the genetic sequences, protein structures, and evolutionary relationships. And why bioinformatics? With the explosion of biological data, because the biological data is vast and tremendous amount of biological data is produced by using the different type of techniques. For example, especially after the Human Genome Project. The Human Genome Project, we all know, is completed in 2003. Okay, and in which the complete genome is sequenced. Okay, the whole genome is completed in 2003 of the human, but we can't analyze it, we can't interpret it because the data is very vast. So, in order to analyze and interpret that data, that sequence can be done by bioinformatics. Traditional methods of traditional method analysis they became inefficient. Okay, because the data is very vast. Bioinformatics provides the tools and techniques to store. The word is store. Okay, store that data, retrieve that data because we don't need the whole data. Okay, we need a specific part of that data. So the retrieval part of the data is also done by bioinformatics. And then when we retrieve the data, we have to analyze it. And this massive amount of data efficiently. Okay, so bioinformatics is used. For example, this is biology. Okay, in which the different things the DNA is available, the plants, the bacteria, viruses, mouse, and different types of data. So we have to analyze it. Okay, and we analyze it by using bioinformatics. Okay, so it's hands on hands in very close to each other. Okay. And what is bioinformatics? The word bioinformatics is it's actually the marriage between biology and computer. Okay. Bioinformatics is the discipline of quantitative analysis. Okay. We analyze quantitatively of the information relating to the biological macromolecules. The macromolecules may be DNA, it may be RNA, it may be with the aid of the computers by using the computers. The development of the bioinformatics as a field is the result of advances in the molecular biology and the computer science. When there was a different advances in the computer science over the past 30 to 40 years, then we have to develop the bioinformatics. The Margaret Dehoff is actually the pioneer of the bioinformatics undertook the first major bioinformatics project in 1965. Okay, they actually developed the first protein sequence database. That name is Atlas of the Protein Sequence and Structure. Now it's actually the PIR. Okay, the key applications: genomic sequencing. First of all, the key application of the bioinformatics is sequencing. Okay, the identifying the DNA sequences, it may be a RNA sequences and protein sequences, and analyzing the genetic variations. Okay, in the genetic variations, it means we have one is the you can say is a reference sequence. Okay, and the others are the individual sequences. Maybe it's a patient or and the other part of the word. We compare the reference sequence with the patient sequence. Okay, and then we check the variations in the sequences. Okay, and that all is done by using the bioinformatics. Okay, the protein structure prediction, understanding the protein folding and function. Okay, proteins is how the protein is fold. Okay, by folding they change how actually they work. Okay, so by understanding the folding of the protein and the functions of the protein is also done by bioinformatics. Okay, the drug discovery, finding the potential drug targets through molecular modeling. Okay, in which the 3D modeling, we also have a complete section on the drug discovery or about the 3D modeling in the upcoming sessions. Actually, we will talk about the genomic sequencing by using the NCBI, the ensemble, the UniProt, and the different types of CAG biogrid. The protein structure predictions also done by using the PDP. Drug discovery by using the Swiss model. Evolutionary biology, in which we check the evolutionary relationships between the different organisms by using the phylogenetics trees. By making the phylogenetics trees, by using the mega is actually a database. It's actually a you can say as actually a software. By using that, we can create the phylogenetics trees. We have a separate session about the phylogenetics, and at the end, the personalized medicine. Now it's actually called the precision medicine. Is the tailoring the treatment based on an individual's genetic makeup. Okay, when we know the genetic makeup of the patient or of the individual, then according to genetic makeup, we tailor the treatments. It's actually done by using the bioinformatics. Okay, now the basic components of the bioinformatics. First of all, the databases. The databases is the basic component of the bioinformatics. Okay, in actually that what is the databases? Is databases is actually uh, it's like a website. Okay, that actually store the biological data. Okay, uh, for example, the GenBank, for example, the NCVI, the Ensemble, the UniPro, the PDP, and the PIR, the different type of switchboard. This type of databases are actually store the biological data. Okay. Now, this is actually the storing the data and retrieving the data is all done by databases. Okay. Now, there are the different types of algorithms and the tools. Okay, that is actually used to perform the sequence alignment. Okay, the one type of the type of the algorithms and tools is sequence alignment. Okay, that is all done by using the cluster omega and blast. In blast, it's actually the about the local alignment, and the cluster omega is all about the global alignment. Okay, in the alignment portion, we will talk about the local and global alignment, and we will also talk about the pairwise alignment and the multiple sequence alignments. And in the bioinformatics, the programming languages is very important. Okay, if you have to know about the Python, R, Perl. If you are interested, I will also make the sessions about the Python, and we have a good knowledge about the Python. And we can also make the separate workshop about the Python, are commonly used in the bioinformatics. And at the end, and the most important is the machine learning and the AI used for the pattern recognition in genetic data. Okay, we have a data. We all have a data. We can retrieve the data. But we have to make the patterns between the biological data. We have to uh, check. We have to compare the data by using the machine learning and AI. Okay, who can learn the bioinformatics? Anyone living in the world. Okay, anyone living in the world having a good knowledge in anything. It may be a physics, it may be a chemistry, it may be a biochemistry, it may be a biology, it may be a statistics, it may be a mathematics, it may be a engineering, it may be a computer science. If you have a knowledge about the any of one of these, you can learn the bioinformatics easily. Okay, because it is an interdisciplinary subject. Okay, so if you are a computer science guy, okay, you have a good knowledge about the computer, okay, but you think you have not good knowledge about the biology, so it doesn't mean you can't understand the bioinformatics. You can understand the bioinformatics. Okay, so anyone with an interest in biology, in the computing, in the data analysis, can explore the bioinformatics. Okay, it is used for the biologists, geneticists, computer scientists, and medical researchers. They all can learn the bioinformatics. Okay, now what are actually the goals of the bioinformatics? Actual, the ultimate goal of the bioinformatics is better understand a living cell. Okay, a living cell, it may be in the human, it may be in the non-humans, it may be in the mouse, in the horses, in the different types of the chimpanzees. Okay, the basic goal of the bioinformatics is to better understand the living cell and not know about the uh, just not about the understanding the living cell, but also how it functions, how the living cell is function is actually the goal of the bioinformatics, and it's actually on the molecular level. Okay, on the molecular level, it means the DNA, RNA, protein level, how the cell is actually wide function. The functions of the cell can be better understood by analyzing the sequence data. Okay, because the flow of the genetic information is dictated by the central dogma. Okay, the word is central dogma. We will talk about it. The central dogma of the biology, in which the DNA is transcribed into RNA and which is translated to the proteins. Okay, the the data and the information is transferred, is flow by from DNA to RNA and to proteins. Okay, so if we know about the protein, the cellular functions are mainly performed by the proteins, whose capabilities are ultimately determined by their sequences. Okay, the cellular functions are actually performed by the proteins. Proteins actually perform the functions. Okay, and the capability of the proteins is actually determined by the sequences. So someone want to. Okay, someone asked, the Naveen Nazar asked if we don't have Python or R programming, we can't we not we do this? You can, but by using the Python or programming language, you can do it even better. Okay, So if you know about if you have that skill, you will be a good bioinformatician. Okay, okay. Actually, the bioinformatics search fields, the first of all, the sequence analysis, the medical bioinformatics, the biostatistics, the machine learning in bioinformatics, the biological data mining, the phylogenetics, the structural bioinformatics, in which the 3D structures of the proteins is actually the one part of the structure. And the gene expression analysis, the computational biology, the genomics, the proteomics, the transcriptomics, the metabolomics, the epigenomics, and at the end, the system biology is actually about the simulation of the server functions. Okay, if you uh, if you know about the one of the these, you will be a good mathematics. You don't need to know about the whole, but if you gradually know about the all the research fields, you will be a good bioinformatician. Okay. Now, what is actually the scope of the bioinformatics? Bioinformatics is actually consists of two subjects. Okay. First of all, the development of the computational tools and databases. Okay. First of all, we have to develop these computation tools and databases. When we develop the computation tools and databases, then what are actually the applications of that tools and databases that we have actually developed, okay, and generating the biological knowledge to better understanding the living systems, okay. So it is actually the scope of the mathematics, like the applications. First of all, the structural analysis, the second one is the sequence analysis, and the third one is the functional analysis, is actually the applications of the bioinformatics. In the structural analysis, the nucleic acid structure predictions like the DNA structure, like the RNA structure, and the protein structures is actually predicted by using the structural analysis of the bioinformatics. And the protein structure predictions, the protein structure classification, like the some structures are like the primary structures, then the secondary structures, alpha helix and the beta plated sheets, and the tertiary structures and quaternary structures. Actually, that are the classification of the protein structures. And at the end, the protein structure comparisons. Okay. And in the sequence analysis, the genome comparison, the phylogenetics, the phylogeny, the gene and promoter prediction, the motif discovery, the sequence database searching, and sequence alignment. It's actually you don't need to worry about what the word is, the motifs, the what the word of the promoters and the genes. You don't need to worry about it because we have a separate section on all about this that of the thing. This session is all about the introduction. Okay, you just know about the words. Okay, you don't need to worry about the what is actually the that words meaning. Okay, in the progression of the that informatics series, we will know about that in a better way. And at the end, the function analysis, the metabolic pathway modeling, the gene expression profiling, the protein interactions, and at the end, the protein subcellular localization. Like this is the actually the cell, okay, and this is the nucleus, for example. And that protein, the specific protein, is where is actually available in the nucleus or in the cytoplasm. If it is in the cytoplasm, in which organelle? It may be in the mitochondria, it may be in the endoplasmic reticulum, okay, and it may be in the lysosome, okay. That's the protein subcellular localization prediction is also done by using the bioinformatics. Okay, now we'll talk about the limitations of the bioinformatics. Okay, as we all know, intelligence is very important in leading to victory in the battlefield. Okay, but fighting a battle like uh, fighting a battle without intelligence is inefficient and dangerous. Okay, if we don't have a intelligence and we just have a numbers of the warriors, but not a lack of the intelligence, it's actually inefficient and dangerous. However, completely relying on the intelligence can also be dangerous if the intelligence is of the limited accuracy. Okay, but the completely relying on the intelligence is very dangerous because maybe the intelligence is have a limited accuracy. Okay, so the over reliance on the poor quality intelligence can yield the costly mistakes if not complete failures. Okay, if the complete failures is not done, it may be costly, can be yield the costly mistakes if we rely on the over reliance, if we rely on the poor quality intelligence. Okay, same like that, the bioinformatics depends on the experimental science. Okay, actually the bioinformatics is the intelligence. Okay, and the experimental science is actually the equality. Okay, it's actually the quantity and it's actually the quality. Okay, so we have to depend on the experimental sciences. Okay, if the bioinformatics is the dry lab, okay, in which we've done all the things by using our laptop or our the computer systems, and in the experimental sciences we have to do the experiments about that data. Okay, we have to done the wet labs. Okay, so we have to depend on the dry lab and the wet lab to understand or to produce the raw data for analysis. Okay, it in turn provides the useful interpretation of the experimental data. Okay, when we get the experimental data, we analyze and interpret the data by using the bioinformatics. Okay, and important leads for the further experimental research. Okay, so bioinformatics is important, but we have to depend on the experimental sciences to do the experiments and conclude some researches. Okay, so this is actually the limitation of the bioinformatics. Okay, what is about the new themes? Despite the pitfalls, okay, there is a different types of the limitations of the bioinformatics, but there is no doubt that bioinformatics is a field that holds a great potential for revolutionizing the biological research in the coming decades. Okay, if we think there are the different limitations of the bioinformatics, so we have to not use the bioinformatics for research. Okay, then we can't update or upgrade our biological research. Okay, then we can't actually analyze and interpret the data. Okay, if we can't interpret and analyze the data, we can't do the research. Okay, so we have to understand the bioinformatics. For example, the very basic example is primate design. Okay, the PCR. Okay, I think so. You all know about the PCRs. Okay, what is PCR? For PCR, we need the primers. Okay, and the primers are always created by using the bioinformatics. Okay, we have to visit the databases, uh, retrieve the data, retrieve the sequence, and then use the primer tree and different types of the tools, and then we create the primers, and then we order the primers by using the different types of the companies available, like the Thermo Fisher and different types of things. Okay, so it is actually the basic example for by using the bioinformatics, different tools and different databases. We design the primers, and after designing the primers by using the dry lab, we shift it to the wet lab, okay, where actually use the primers. So we actually highly need bioinformatics, and it's a very important. And there is a lot of scope about the bioinformatics. If you are a bioinformatician, you can you can get the salary, double salary of the others that do not know about the bioinformatics, but is actually a scientist. Okay, a scientist if gets like the $100, and the bioinformatician if have a good skills can earn the $200 per month. Like that, it's an example. Okay. There is no doubt that bioinformatics is a field that holds the great potential for revolutionizing the biological research. Okay, to gain a deeper understanding of the cellular functions, if we want to know about the cellular functions, the mathematical models are needed. Okay, we have to know about the mathematical models to simulate. Okay, we have to simulate the functions and a wide variety of the intracellular reactions and interactions at the whole cell level. These molecular simulations of the all cellular processes is termed the systems biology. It's actually the systems biology in which we simulate the cellular functions and the cellular processes. Okay, now this is actually the end of the presentation. Then we move to the towards the databases. Okay, the final thought is the bioinformatics is the revolutionizing how we understand life. Okay, we understand life by using the bioinformatics at the molecular level. Okay, whether you are a student or a researcher or an enthusiast, learning the bioinformatics opens the doors to exciting discoveries in the genetics, in the level of the medicine, at the level of the biotechnology. Okay, now if you have any questions about that presentation or all about the limitations of the scopes and the goals of the bioinformatics and the different types of the applications, if you have any question, you can ask by using by opening the mic or by using the inbox. Okay, Asim Malik, you raise hand. Do you have a question? If anyone have question, can ask me by using mic, by opening the mic or by using the message.
No, Sunny.
Max, you have a question? Do you have a question?
No, I don't have.
Okay, okay. Mohammed Bakir, do you have question? We Nazar Asim Malik, do you have question? You can ask me. If not, I am going to towards the databases, the basic, the generic overview of the databases. Because in the next session that is all about the databases, and then we progressively move towards the upper level of deep informatics. Okay, I think so. No one has questions, so we will move towards the uh, introduction of the databases. Okay, I am going to share the screen to all of you again. Okay, do you see the screen? Do you hear me? Do you see the screen?
Yes, yes,
Okay,
Yeah.
yes.
okay. Uh, now I am using the different types of the databases. but in that session, I'm using the NCBI. Okay, if you have laptop in front of you, you can also open the NCBI and do what I am also doing. Okay, if you have you can practice what I am doing. Okay, okay. First of all, the generic database of the NCBI is N of the bioinformatics is NCBI. Okay, it's actually the National Center for Biotechnology Information. Okay, that has a lot of data about the DNA sequences, the RNA sequences, and the protein sequences you can get from the NCBI. Okay, just click on the National Center for Biotechnology Information (NCBI). This is actually the front of the website. Okay, this is actually the window. Okay, now if we check that, welcome to the National Center for Biotechnology Information, in which we can submit the sequences. Okay, if we have a sequence about any patient, you can submit here. You can download the different data. You can also learn about the documents and the different types of the. You can also check the tutorials about it, and you can also develop the APIs and code libraries. We can in the next session we also develop the codes and the APIs. Okay, and by this we can also submit the different types of data and analyze. Okay, this is actually important. We can analyze that data by using the NCBI. And at the end, the research. NCBI use the collaborative projects in which you can collaborate different types of projects and interpret and analyze the data. Okay, NCBI had different types of the resources like the PubMed, in which we can download the different types of research papers. The bookshelf is also same. The PubMed Central is also you can say the kind of the same like the PubMed and the BLAST. In the next session, we can talk about the sequence analysis. Okay, I think so. Third session is about the sequence analysis. Okay, and nucleotides, the genome, the SNP, the single nucleotide polymorphisms, the gene, the protein, the PubMed, PubChem. It's actually the popular resources of the NCBI. Okay, now we click on that. It's actually about the databases present in the NCBI. Okay, NCBI is like a primary database. Okay, in the next session we talk about the kinds of the types of the databases. The first one is the primary databases. The second one is the secondary databases. The third one is the tertiary or the specialized databases. The NCBI is a primary database. Okay, in that databases, the different types of the secondary databases or the tertiary database, the special databases are available. Okay, like the assembly. Okay, in which you get the DNA assemblies. Okay, the bio collections, the bio project, the bio samples, the different types of books you can get from this by using the NCBA. The ClinVar, in which the clinical variations, the different types of diseases is quoted in that database. And the conserved domains, there are different types of domains. Okay, the domains is actually the part of the proteins. Sorry, it's the actually the parts of the domains independent. Okay, and dbcap, dbvar, gene. Okay, uh, uh, gene, genome, and different types of things. Okay, we click on N and nucleotide. Okay, we will talk about the nucleotides. Today we talk about the nucleotides, and we search here NG. Okay, if you have the laptop in front of you, you can search about energy. Like one is we can say is the breaker one. Okay, and after writing about the G, click on search. Okay, after clicking on search, you can get that type of results. Okay, the breakup one is present in different recipes. Okay, like it is present in animals. Okay, it's also present in plants, in the fungi, the protista, the bacteria, the archaea, the viruses, and you can also customize here. There are different types. You can also unclick or click, select or unselect it. The molecular type, it may be a genomic or DNA sequences or data, messenger RNA. Sources, it may be from the GenBank or the reference sequences. We'll talk about that GenBank. GenBank is actually the generic form of the NCBI, and the RefSeq is actually the reference sequences. Okay, that sequences that are experimentally verified. Okay, are all present in the ref sequences. Okay, when we count the 41,000 and we will count it's almost the 45. Okay, let's say that are the 45,000 sequences. Okay, in which out of 45,000 sequences, the 31,000 sequences are actually verified. Okay, that is why it's present in the ref sequence. And the sequence type, it may be a nucleotide sequence, it may be a ESTs, it may be a GSS. Okay, there are different types of the sequences. And the genetic compartments, it may BRCA1 is present in the mitochondria. We also know that the uh, application of the bioinformatics in which we can know about the subcellular localization of the proteins or the genes. Okay, so in the genetic compartments, we can say the mitochondria. In the mitochondria, the BRCA1 is present. Okay, and it's also present in the plasmids. Okay, the sequence length may be different. You can also select it. Release data, you can also select it. Okay, now I am going to results by the taxon. Okay, in which is actually about the taxons. Okay, you can also click on the that like the home in the Homo sapiens is actually present in Homo sapiens in the musculus in the present synthetic construct. Okay, you can also click on the tree. When you click on the tree is made. Okay, like in the eukaryotes, you can select it in the animals. First of all, we all are eukaryotes. Okay, so we in the eukaryotes we are animals. Okay, in the animals we are chordates. In the chordates we are vertebrates. Okay, then we click on the more. Okay, when we click on chordates, there are different types of lanceolates and the tunicates. Okay. Now we can select here different types of assemblies present here. Okay, we I am going to click on the Homo sapiens. Okay, we are all are humans. Okay, now we click on Homo sapiens. After clicking on Homo sapiens, that type of data is come in front of us. Okay, the data is like uh, it's like a flat file. Okay, in front of us is the flat file. And after clicking on the fast sequence, I am opening in the new tab, and you can also check. Okay, I am not going to give you survey. Okay, in this that sequence we use for making the primers for primer designing. We use that sequence. Okay, if we want to uh, analyze the BRCA1 gene present in our patient's sample. Okay, so we have to design the primers by using that sequence. Okay, in the first sequence, the first one is the definition of the sequence. Okay, it's actually NC. That number is actually the accession number. Okay, the unique identifier of the DNA or of the gene. Okay, and the next one is starting from that is the sequence. Okay, you can click. You can also copy it and you can also download it by click on send. It may be a coding sequence. You can also click on the gene features and the complete code. Okay. Then we click on the file. Okay. Then we click on the fastest sequence or the different types of summary, but we click on the fastest and create file. Okay. Okay. Now we can download it. Okay. The sequence is download. And in graphic sequences, you can check the where is actually the BRCA1 is present. Okay. Like that, if the whole is present is actually the chromosome. Okay. In the chromosomes, the BRCA1 is present here, like that. Okay. The BRCA3 is present here. The CA10 is present here. The ASIC2 is present here. Okay. The sequence is downloaded. We just open it. Okay, you can get the data from it. Okay, the first of all is the locus. Okay, the where is the actually the that gene is present. Okay, the accession number. First of all, the accession number. This is actually the unique number of the unique identifier of the BRCA1, and the how much base pairs are present in. It's like in the millions, and it's actually a molecular type is DNA, RNA, or a protein. It's actually a DNA, and it is a circular or linear because we are in the humans the nucleic acid is present in linear form, so it is a linear. And con is actually a coded, so the Homo sapiens we use the con. And when this sequence is updated on 26th August 2024. And in the definition, we get the summary of the definition, like the Homo sapiens. They isolate the CHM13 chromosome 17, alternatively assembly T to T, CHM13V 2.0. Oh, actually, it is the definition. And the accession number, this is actually the accession number. And the version. Version is a same like the accession number, and at the end is the how much time it's updated. Okay, it's a one, it means it's not updated, it's not changed. Okay, so the version number is one. If it's changed one time, the version number is upgraded to the two. The DB link is actually the databases in which the that sequence is present is also present in the bio project. You can click on it and get the results and the bio samples and assembly. The keywords and the source is actually the Homo sapiens and the organism. In the organism, it's actually the taxons. In the eukaryote, metazoa, chordata, craniator, it's actually the you can say the top down of the taxon. Vertebrate, uterostomy, mammalia, eutheria, Eurocondoglaris, primates, Haplorhini, Cattlorhini, Hominidae, and the Homo. Okay, at the end is the Homo. Start from the eukaryotes and at the end the Homo. Oh, we can also get the taxons. And in the comment about the identifications. Okay, you can get the when the assembly is done. Okay, and at the end is all about the features. Okay, the where is actually the protein is present. The BRCA1 gene is actually transcribed into RNA, and that RNA is translated into DNA. Oh, sorry, translated into protein. Okay, that protein is present, like the that sequence is present in female. Okay, and the tissue type is hydatidiform mole. Okay, and that cell line is the haploid cell line. Okay, by using bioinformatics, we can get different types of, we can get the knowledge about the sequence. Okay, we can interpret it, we can use it. Okay. Now I am going to talk about the blast. Okay. In the next section, we talk about the whole things in the progressive way. But now I am just going to introduce the some types of blast. Okay, there are different types of blast like the nucleotide blast, the blast X, the T blast, and the protein blast. But I'm using the nucleotide blast. Okay, like that we download the sequence. Okay, and I'm using that sequence for using blast. Okay, like that we can enter the query sequence. Okay, by using blast uh, we can compare the different types of sequences. Okay, like in that sequence is actually the complete the reference sequence. Okay, I'm going to change the sequence from here like that. Okay, okay, close the program. Okay, like that. I am just going to get the sequence again. Just click on it. Okay, like click on the first sequence. We can get the first sequence. Okay, like that. I am just going to uh, give you the intro introduction of the blast. Okay, I am just going to copy it. Okay, Control C, like that, copy it. And okay, it's actually the same sequence. But I'm going to change that T with A. Okay, I'm just changing the T with A and not changing the anything from this. Okay, you can also change the like the organism. You can exclude the organism. You can add the organism. Like you can add the organism like the Homo sapiens or the different type of chordates and eukaryotes like that. Okay, and just click on the blast. It's actually align the sequences. Okay, it's actually compare the sequences with the reference sequence. Like I'm going to change the sequence. It means we change the sequence with the someone like the patient sequence. Like the no similarity is found. Okay, the we're going to select the long sequence like that. Okay, we change the sequence and we change with A. And uh, this is using the machine learning and different types of tools and algorithms and give the comparison between the reference sequence and the sequence that we give. Okay, the 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 sequence that we give is actually the query. Okay, and the result we get is actually the that result. Okay, it's actually present in the Homo sapiens. Okay, the sequence is same like that. Okay, Homo sapiens, that is the complete sequence. Okay, Okay, the percent of the identification is 96% because we change the sequence with C with A. Okay, that is why the percent of the identification is changed. The E value is 0. The E value is actually the random sequencing. Okay, it must be 0. If it change, it means the sequence alignment is not done well. Okay, the query we give is actually completed 91%. Okay, now we click on the graphic summary in which the long red line is actually good. It means the sequence is aligned well. In that way, the sequence is not aligned well. And in the alignments, we get where is actually the changes. I'm going to change by a pairwise or the dots. I'm going to change with the pairwise with the dots. Like by changing just one sequence from the one nucleotide, this is actually the problem is here because the frame shift is done. Okay, due to frame shifting, the sequence is changed. Okay, the sequence like that there is a C. Okay, in the query is C, but in the reference sequence there is a T. Okay, that is why the patient sequence is different from the reference sequence. Okay, okay. Thank you so much for being part of this wonderful journey. In the next session, we will talk about the databases in the depth, in the good way, in like all about the databases. The next session is all about the databases, and the third session is all about the sequence alignment. The fourth session is all about the multiple sequencing and global and local sequences. Okay, the fifth session is all about the protein modeling, the Swiss model, and the different types of modeling, and the 3D structure of the proteins. And the fifth, sixth, fifth and sixth session is all about the phylogenetics, and the eighth session is all about the interactions and the pathways. Okay, so in that way, the sessions are going, and you can become the pro in the bioinformatics. And we will have a good, I am going to also include the Python language in which you can learn the Python by me. Okay, thank you so much for being part of this. Hope so you understand the first session of the bioinformatics. Well, if you have any
Thank
question
you.
about today, you can ask me.
I
Nvinder, you
think
can also contact me.
you will share the slides, yeah?
Yes, yes.
Okay, thank you very much.
I share the slides and other data, the exclusive data to you, all of you, uh, by using the, WhatsApp. Okay.
Yeah,
If you have any question, you can ask me here. Yes, you can also ask me. Asim Malik, you can also ask me any question you can have. Ask me.
no question for now.
Okay, okay,
Thank you very much.
okay. Do you enjoy the today's session? Yes. Do you enjoy today's session because other ones are left? Just you are here.
I didn't understand. What do you